I can get started. So hi, everyone. Thanks for joining today. I'm Jason Key at SB Grid, Boston, Massachusetts. If you have questions during the talk, feel free to put them in the chat. You can send those questions here and we can unmute you at the end and you can ask your question. And with that, I'll turn it over to Cynthia Wolberger for the intro.
Great, thanks. Hi, everyone. So it's really a pleasure for me to introduce Evan Worden, who's a postdoctoral fellow in my lab. So Evan got his undergraduate degree at Whitworth University, and he moved to Berkeley, where he got his PhD doing work in Andy Martin's lab on the coupling between substrate ubiquitination and proteasomal degradation. And there he did a beautiful biochemistry and some X-ray crystallography. I was fortunate enough that he moved to do his postdoc in my lab, where he's been talking about he's been working on what I'll talk about today, on his work on crosstalk between histone ubiquitination and methylation. And in the process, he's really become an expert in cryo-electron microscopy. Evan's work has been recognized already by numerous awards and fellowships. So he had an NSF predoctoral fellowship. He won the Cosarelli Thesis Prize at Berkeley. And as a postdoc, he's a Damon Runyon Cancer Research Fund Fellow, and recently won the Johns Hopkins Young Investigators Day Paul Ehrlich Award. And what you'll hear from him is the beautiful work that he's done since joining my lab. That's been already recognized. So the title of his talk is Mechanisms of Crosstalk Between Histone Ubiquitination and Methylation. So take it away, Evan.
Okay, thank you so much, Cynthia. Hopefully everyone can hear me. But first, I want to thank again the SB Grid seminar series for the invitation, and also thank you to my boss, Cynthia, for letting me take over this talk and get to talk about some of the work I've been doing on trying to understand how crosstalk is regulated between histone ubiquitination and methylation. So in eukaryotic cells, DNA is compacted and stored by being wrapped in octameric core of histone proteins. And the complex between DNA and this histone protein octamer is called the nucleosome. And in addition to storing our DNA, our histone proteins are also extensively decorated by post-translational histone modifications. And these modifications include methylation, acetylation, phosphorylation, and also ubiquitination, among many others. And each one of these individual histone proteins can be modified with multiple modifications. And then the combination of these histone modifications can then instruct the cell to either activate or silence certain genomic loci or recruit certain proteins to genomic loci. And the combinatorial interpretation of this histone modification landscape is called the histone code. And there is just a huge variety of diversity in the kind of modifications that are found on histone proteins. And these can range from acetylation to phosphorylation, like I described on the previous slide, but there are also larger complex marks such as ADP ribosylation and ubiquitination. And seemingly, new modifications are found every year, shown by the ellipsis here. So this modification landscape is already quite astounding and complicated, but it's getting more and more complicated every year. And in addition to the large number of histone modifications, there is also a huge variety in the chemical complexity of these histone marks. And you can see that clearly if we compare one of the simplest marks, namely this methylation of a lysine epsilon amino group, which just attaches a single CH3 to a lysine side chain. This is a very small modification, very simple, but you can also have very complex marks attached to histone proteins. And that's exemplified by the attachment of the entire protein ubiquitin to the lysine side chain. So there's this huge variety, both in number and chemical complexity of these different histone marks. Now, in eukaryotic cells, all processes that require access to DNA are regulated by these post-translational histone modifications. And very broadly speaking, these histone modifications can be grouped into silencing marks, which are found primarily in heterochromatic regions where the DNA and nucleosomes are highly packaged and very little transcription occurs, and activating marks, which are found in euchromatic regions where the nucleosomes are more well spaced out in this beads on a string type arrangement, and active transcription occurs in these regions of the genome. And one very important activating histone mark that is found in these euchromatic regions is the non-degradative mono-ubiquitination of histone H2B. And this ubiquitination modification is sort of depicted here, where we have a structure of the nucleosome shown here, and then attached to this red histone protein H2B is a ubiquitin molecule. Now, histone H2B ubiquitin is a very unique histone mark because in addition to modifying the histone it's attached to, this histone mark also stimulates the deposition of two other histone marks on a separate histone protein, namely histone H3. And in particular, H2B ubiquitination stimulates the deposition of methylation on H3K79, and it also stimulates the deposition of methylation on lysine 4 of histone H3. And this process by which one mark stimulates the addition of another mark is called this histone crosstalk, or in the case of this between histone proteins, it's called trans-histone crosstalk. And the crosstalk between these ubiquitin marks and these methylation marks was elegantly described all the way back in 2002 by multiple different groups. And what I've shown here is just some of the original data showing that in, for instance, for H3K79 methylation, in wild-type yeast cells, you get robust H3K79 methylation. But when you delete the ubiquitination machinery that is responsible for attaching this H2B ubiquitin mark, you completely ablate the H3K79 methyl mark. And that's recapitulated completely also with H3K4 methylation in yeast, where the H3K4 methyl mark is completely dependent on ubiquitination of histone H2B. So in addition to stimulating the deposition of H3K9 and H3K4 methylation, H2B ubiquitination also directly templates these methyl marks onto active genes. And so what I have depicted here is the chromatin state of your favorite active gene. We have Pol2 positioned at the transcription start site, ready to initiate transcription. And once transcription occurs, the addition of H2B ubiquitination is believed to occur co-transcriptionally with Pol2. And if you look at a metagene profile of the H2B ubiquitin signal on these genes, you'll see that ubiquitination of histone H2B occurs right after the transcription start site, and then it slowly decreases over you go through the body of the gene. Now, if we compare the metagene profile of H2B ubiquitination with H3K4 methylation or H3K79 methylation, we can see that both of these methyl marks peak right after the transcription start site, but H3K4 methylation peaks to a higher extent and then decreases much more rapidly than H3K79 methylation, which has a more soft peak and a longer decrease. So yeah, this H2B ubiquitination is important for deposition and also templating these methyl marks onto genes. So the similar profile of H3K4 and H3K9 methylation on genomic loci implies that these two methyl marks are probably very closely related in their function. And indeed, if we look at the different functional characteristics of these two methyl marks, we can see that, of course, they're both completely dependent on H2B ubiquitination. And both H3K79 and H3K4 methylation are both associated with active transcription. However, they also have some divergent functions, where H3K79 methylation, in yeast at least, is associated with telomeric silencing. And in H3K4 is associated with different kinds of cell differentiation. H3K79 methylation is important for the DNA damage response, and H3K4 methylation is found at enhancer regions. And finally, both of these different methyl marks, when they're dysregulated, have very important roles in the development of certain cancers. So these divergent functions of H3K79 and H3K4 methylation may be in part a product of structural differences between these two marks. So if we go ahead and look at the structure of a nucleosome, we can begin to understand how these two marks are chemically different in the context of the nucleosome. So H3K4 lies on a flexible histone tail of the nucleosome. And you can see that here, this red spot here is this H3K4 side chain, and it's on this flexible tail that comes out of the nucleosome. But H3K9 is actually not located on a histone tail, but it's located in the folded core of the histone octamer. In the case of H3K4, this side chain is very, very far away from ubiquitin. And this ubiquitin is supposed to stimulate the deposition of a methyl group on this lysine. So it's a very, very far distance to get past. But in the case of H3K79 methylation, this side chain is actually very close to the ubiquitin. In H3K4, because this side chain is on this flexible tail, the H3K4 side chain is actually very highly accessible. But because H3K79 is located in this folded histone core, the side chain of this residue is very inaccessible for modification. And finally, the proteins that are responsible for attaching these respective histone marks are highly different. So H3K4 methylation is catalyzed by the COMPASS complex in yeast, which is a 400 kilodalton multi-subunit complex that contains a set domain methyltransferase. However, H3K79 methylation is only methylated by a single protein, this Dot1L protein, which has a completely different catalytic fold from COMPASS in the Set1 catalytic domain. So it's really clear that these two methyl marks are highly different in their structure and chemical environment, and also are methylated by highly different enzymes. So at the beginning of my postdoc, it was really not clear how ubiquitin is able to stimulate the methylation of these two marks by activating these two highly divergent proteins. And so it wasn't really clear, even from the structures, where ubiquitin might bind these proteins. There was no real ubiquitin binding motifs that could be identified in these proteins. And because of that, it wasn't clear what parts of the protein even interacted with ubiquitin. And as I mentioned, both Dot1L and Set1, the catalytic subunit of COMPASS, have these completely different methyltransferase folds and are of completely different families. They're unrelated, yet they have this similar stimulation mechanism by ubiquitination. And also paradoxically, it had been shown biochemically that ubiquitination of histone H2B does not actually increase the affinity of these enzymes for the nucleosome. And so it wasn't simply some sort of recruitment mechanism that could explain this activation by these two enzymes. So at the start of my postdoc, I really wanted to understand the molecular basis for the crosstalk of this single ubiquitin mark and these two highly different methyl modifications. I wanted to understand what the molecular basis for this crosstalk was. And to begin answering that, I started working on the H3K79 methyl mark. So as I mentioned, the protein that is responsible for methylating histone H3K79 in the nucleosome is called Dot1L. And Dot1L itself is a very long protein with an N-terminal catalytic domain and a positively charged DNA binding sequence that just has a bunch of lysines and arginines. And this combination of Dot1L catalytic domain and the DNA binding region is actually constituting a minimal fragment that is required for activity on nucleosomes. C-terminal to this minimal fragment, Dot1L has landing pads for various interaction partners, and these interaction partners are responsible for recruiting Dot1L to different genomic loci in the cell. However, as I mentioned before, even though we've had a structure for many years, it was not clear at all, either from the primary sequence or the structure of Dot1L, where ubiquitin could bind, because there were no known ubiquitin binding motifs on this protein. And so to begin to understand how Dot1L is stimulated by H2B ubiquitin, I used this minimal fragment of Dot1L and determined a Cryo-EM structure of Dot1L bound to a ubiquitinated nucleosome. And I was able to resolve this structure to around 3.9 angstrom resolution. And as we can see, and this is very common with nucleosome binding proteins, the center of the nucleosome has a very, very high local resolution, and the side chains and backbone atoms are easily built into the density here. But any peripheral binding proteins are much less well resolved than the nucleosome's core. However, the resolution of this structure was definitely good enough to unambiguously dock crystal structures of the nucleosome and ubiquitin and Dot1L into the EM density. And from the structure, we can clearly see that ubiquitin directly binds to Dot1L in this C-terminal section of the protein on this side of the nucleosome. And the N-terminal part of Dot1L is actually lifted up over the nucleosome on the other side. Now, if we move around the structure, we can see that all the features of the EM density are accounted for by the published crystal structures of Dot1L and the nucleosome. And we can see that ubiquitin actually interacts with Dot1L using a conserved hydrophobic surface that is centered around leucine 73, 71, and isoleucine 36. And this interaction surface of ubiquitin is actually very distinct from a more canonical interaction surface of ubiquitin that occurs around isoleucine 44. And a huge majority of ubiquitin binding proteins actually utilize isoleucine 44 for binding. But Dot1L is utilizing this separate interaction surface, and isoleucine 44 is not contacting Dot1L at all. And this is actually consistent with prior biochemical experiments showing that this residue is not important for Dot1L stimulation. And if we look at the interaction between ubiquitin and Dot1L, we can see that this hydrophobic patch of ubiquitin binds in a somewhat unexpected manner to Dot1L, where Dot1L interacts with these hydrophobic residues using a hydrophobic surface on its C-terminal section composed of these residues in green. And importantly, this hydrophobic surface of Dot1L does not really resemble any known ubiquitin binding motifs. And so it's therefore a sort of novel ubiquitin binding motif that is specific for Dot1L. And importantly, again, this binding surface of ubiquitin had been previously identified using biochemical screening, where Tom Muir's group in 2015 identified that these two leucine residues, when they are mutated to alanine, you completely lose Dot1L activity. So the interface that we found in our structure is in very close agreement with prior biochemical results. So even though this structure nicely showed how Dot1L interacts with ubiquitin, it actually didn't reveal how Dot1L is activated by ubiquitin. And that's because the structure that I determined here is actually Dot1L held in an inactive state. And you can actually clearly see that when you just compare the distance between a modeled SAM cofactor in the Dot1L binding site and the H3K79 side chain. And so you just look at the distance between these two groups. It's 21 angstroms away. And this is a very, very large distance on the atomic scale. And it's very unlikely that some methyl group is going to launch itself from this SAM methyl donor and somehow fly all the way over to H3K79 and land on this epsilon amino group. So obviously, that's not going to happen. So we termed this inactive state of Dot1L a poised state, because we think that this state of Dot1L exists probably either right before or right after the methyl transfer reaction has occurred. And it was clear from the distance between the SAM cofactor and the H3K79 side chain that something would have to close this gap in order for catalysis to occur. So to try trapping Dot1L in some kind of active state where it's much closer to the H3K79 side chain, I took advantage of the fact that set domain methyltransferases have been known for quite a while to bind very tightly with 10 to 1,000 times higher affinity to mutations of their substrate lysine to norleucine. And norleucine is a non-native amino acid that is very similar in structure to lysine, except that it does not contain the epsilon positively charged amino group. And so it's just an aliphatic side chain. And so I went ahead and prepared nucleosomes containing an H3K79 norleucine mutation. And I wanted to see if Dot1L bound more tightly to nucleosomes containing this mutation. And so to test if Dot1L actually bound these nucleosomes tighter, I did a very simple gel shift experiment looking for the ability of Dot1L to shift a nucleosome band to some sort of higher mobility. And I found that when you compare the wild type nucleosome with a H3K79 mutated nucleosome, the two nucleosomes bind to Dot1L with almost identical affinities. And if you look very closely, the one indication that maybe Dot1L binds more tightly to this nucleosome with the norleucine is a slight premature shift of this band at 2 micromolar Dot1L. And when I saw this, I thought, well, this is definitely not 10 to 1,000 times higher affinity, this mutation here. And so I didn't actually think that this would work, but I had time coming up on a microscope. So I went ahead and prepared complexes containing this norleucine mutation and sent those samples off to the NCI Cryo-EM Center. And I got back a nice Christmas gift because from that data set, I was able to determine 2 structures of Dot1L bound to a nucleosome in the active state. The first structure contained a 2-to-1 complex where 2 Dot1L molecules are bound to a single nucleosome, and that structure resolved to around 3 angstrom resolution. And it also determined a second structure of Dot1L bound in a 1-to-1 orientation with the nucleosome, and that structure resolved to around 3.5 angstrom. And the EM density of these maps was very good, and we were able to clearly resolve all the side chain features of Dot1L and ubiquitin and the entire nucleosome. And to determine this 2-to-1 structure, we actually used a 2-fold symmetry to get to this 3 angstrom resolution because this is a 2-fold symmetric binding interaction. But for the 1-to-1 complex that was at 3.5 angstrom, we did not impose any symmetry. And aside from the fact that one structure contains 2 copies of Dot1L and the other structure only contains 1 copy, there are actually very few differences between these 2 structures. However, one small difference between the structures is actually in this N-terminal extension of Dot1L, which is much less resolved in the 2-to-1 binding structure than it is in the 1-to-1 binding structure. And we think that this weak density in the 2-to-1 binding orientation is probably due to the imposed 2-fold symmetry, where this N-terminal domain may not be as well correlated with itself on the opposite side of the nucleosome. But nevertheless, the 2-to-1 structure of Dot1L bound to the nucleosome did have excellent density, and we were able to see many important interactions, including interactions in the Dot1L catalytic active sites, Dot1L interactions with ubiquitin, and of course, excellent density within the nucleosome itself. So if we compare the active state structure with the poised state structure, we can see that in the active state structure, the N-terminal part of Dot1L is actually sitting down and contacting the nucleosome. But when it transitions to the poised state structure, this part of Dot1L lifts up off the nucleosome and is far away from the H3K79 side chain. And this transition from the poised state to the active state actually occurs around a pivot point. And that pivot point includes this ubiquitin attached, the ubiquitin attached to histone H2B. And if we zoom around one more time, we'll see again that this transition here occurs to the poised state around this ubiquitin residue and then comes back down and contacts the nucleosome in the active state. Now in the active state, I saw basically all the same interactions between Dot1L and ubiquitin that I observed in the poised state. And we also saw the same interactions between Dot1L and the acidic patch shown here. But in the active state, there are also several more interactions that occur between Dot1L and the nucleosome. For instance, Dot1L forms direct interactions with the histone H4 tail and also with the H3K79 substrate lysine. Now this acidic patch contact here is actually a very common interaction hub of nucleosome binding proteins. And this is because this is a distinct charged surface of the nucleosome that many different proteins utilize to interact with the nucleosome. And in the case of Dot1L, the interaction with the acidic patch is actually mediated by a single arginine residue in Dot1L, this R282. And if you compare the active and poised states, so the poised state is here in yellow and the active state is here in green, the only residue in this entire structure that doesn't move between the two states is this arginine residue. So the actual pivot point transition between the active and poised states is this arginine 282 residue. And this interaction mechanism where a single arginine interacts with the acidic patch is actually a very common mechanism of interaction with nucleosomes. And this arginine interaction mechanism has been seen for multiple nucleosome binding proteins, including the SAGA-Dub module, PRC1, RCC1, and the SIR3-BAH domain. So this is a very common mode of interaction with the nucleosome. So in the poised state structure of Dot1L, and in fact, in every crystal structure of the nucleosome that has been published, H3K79 is sort of lying flat down along the surface of the nucleosome here. And in this orientation, this H3K79 side chain is inaccessible. Now, when I saw how inaccessible this side chain was, I was sure that in order for Dot1L to access this side chain, there would have to be some massive conformational rearrangement within Dot1L. And so I really fully expected Dot1L to somehow unfold and sit down on the nucleosome in order to access this H3K79 side chain. However, when we determined the active state structure, we found that Dot1L doesn't actually change conformation at all. It's almost completely superimposable with published crystal structures. And instead of Dot1L changing conformation, the H3K79 side chain goes through this huge conformational rearrangement that changes the backbone and side chain conformation of H3K79. That lifts this side chain from this inhibitory inaccessible conformation to a 90-degree rotation that sticks it straight up into the Dot1L active site. Now, we were just blown away by this conformational change within H3 because nothing like this had been described before. And it's important to realize that this isn't just some sort of rotameric switch of the lysine side chain, but this is actually a complete disruption of the secondary structure around H3K79 that includes disruption of the hydrogen bonding network in a histone helix that directly abuts histone H3K79. And this is a very energetically costly disruption in the structure of histone H3. And it was a very unprecedented finding, and it was the first example of a core histone fold being actively disrupted during catalysis by an enzyme. Now, the outcome of this conformational change is that this H3K79 side chain comes within striking distance of the SAM cofactor. Now, we don't actually have a H3K79 in this structure because this was a norleucine mutation that allowed us to capture the state. But when I modeled in a lysine side chain here, it seems like a lysine here would come within 3 angstroms of this SAM cofactor, which is now close enough to support methyl transfer. And if I build in the rest of the Dot1L structure around this H3K79 side chain, you can see that the altered conformation of histone H3K79 is stabilized by contacts with 2 important Dot1L residues, this F131 residue, which comes in underneath and scoops H3K79 out of its inhibited state, and this tryptophan 305, which braces one side of the lysine and stabilizes it in the active site. And importantly, mutating either of these residues completely kills activity. And very surprisingly, we also discovered that there's a third stabilizing interaction that can be seen. And this is a stabilizing interaction that is donated from the histone H4 tail with this arginine 19 subunit or amino acid. And importantly, the electron density in this section of the map was very good, and we could clearly see all these different stabilizing interactions within the active site density. Now, the participation of the histone H4 tail in formation of the active Dot1L complex was particularly interesting because it suggested that the nucleosome itself might help stabilize H3K79 in the altered conformation. And this structure showed that this histone H4 tail actually binds within a shallow groove on the surface of Dot1L. And this was a completely unidentified groove because this groove is actually closed when Dot1L is not bound to the nucleosome. And if we zoom in on this part of the structure, we can see that these histone H4 residues make multiple contacts with Dot1L and histone H3 within this groove. As I just mentioned before, this histone H4 R19 residue directly contacts the backbone atom of H3K79, which stabilizes this H3K79 side chain in the active conformation. And in addition to this R19 residue, a second interaction occurs between this R17 of histone H4 and an acidic pocket within Dot1L. And it's very likely that this R17 is interacting with conserved acidic residues in this pocket. And importantly, both of these interactions agree with prior biochemical studies that identified the histone H4 tail as an important interaction site for Dot1L. And you can see here that mutation, this is by Fingerman in 2007, either mutation of this R17 contact or this R18 contact both completely ablate the ability of Dot1L to di and trimethylate H3K79 in yeast, whereas wild type Dot1L is able to robustly methylate H3K79 in yeast. So therefore, it appears that once Dot1L induces this conformational change in histone H3 and breaks these hydrogen bonds in this H3 helix, it needs this H4R19 residue to come in and stabilize this new conformation of H3K79. And I like to think of this as a kind of substrate-assisted catalysis, where this important altered conformation of histone H3 is stabilized by other contacts within the substrate that allow catalysis to occur. So if we take a step back again and look at the full active site, we can really appreciate all the different stabilizing interactions that occur between Dot1L and histone H4 that force this H3K79 side chain into a new conformation and allow catalysis to occur. And the end result of all these stabilizing interactions is that the H3K79 side chain is inserted, oops. No, this video is not playing, but this H3K79 side chain gets inserted deep into a hydrophobic tunnel that allows it to access this SAM cofactor. And now that we have the full view of the Dot1L active site, we can begin to answer one of these long-standing questions, which is exactly how Dot1L is able to catalyze methyl transfer. And one of the important steps of the methyl transfer reaction is this deprotonation of the lysine side chain. But in this Dot1L active site, there are actually no ionizable residues that could extract this proton from the lysine. And so what we think is going on is that just forcing this lysine into this hydrophobic environment is lowering the pKa of this epsilon amino group enough to allow it to target and transfer the methyl group. Okay, so we now have a pretty good idea of how Dot1L is able to be stimulated by ubiquitination of histone H2B. What we think is going on is that ubiquitin is able to restrict the movement of Dot1L on the nucleosome, and it also acts as a pivot to help Dot1L switch between these poised and active states. And then once Dot1L lands on the H3K79 side chain, it forces it into this new conformation, and that new conformation is then stabilized by H3K79, or by histone H4. And we like to think of this as a bird looking for a worm, where in the poised state, Dot1L is looking for a worm, and then in the active state, it is ripping up this H3K79 side chain. So what about the other methyl marks? So there are two methyl marks I was really interested in. One of them is H3K79, but the other is H3K4. And H3K4 methylation is stimulated by H2B ubiquitin. And the protein complex responsible for this methyl mark is the COMPASS Set1 complex. So COMPASS is an 8 subunit complex, and the catalytic subunit of this complex is Set1, which has this requisite set catalytic domain. And previous work by many different labs has shown that not every single COMPASS subunit is required for activity, and that there is actually a minimal core subcomplex of COMPASS that can be fully active and stimulatable by H2B ubiquitination. And this is a very conserved subcomplex of COMPASS. And I just have here some of the human homologs of the yeast counterparts. And these human homologs are found in the related human Set1 complex and also the ML core complex that is another human H3K4 methyltransferase. Now, just before I started this project, 2 wonderful structures came out from the Skiniotis lab and from Ning Zheng's group in 2018. And these papers revealed the structure of the isolated minimal ubiquitin sensing COMPASS complex. However, it was not clear at all from these structures how this COMPASS complex is able to recognize ubiquitin or interact with the nucleosome. So I used the same norleucine replacement strategy I used for Dot1L to determine a structure of the yeast COMPASS complex bound to a ubiquitinated nucleosome. And this structure I was able to resolve to around 4 angstrom resolution. And again, we see this all the time with nucleosome-bound proteins, where the nucleosome is very high resolution, and then the resolution of things binding to the nucleosome sort of decrease as you reach the periphery. So the Cryo-EM density of this COMPASS structure allowed me to dock in and rebuild structures of the yeast COMPASS. And we were actually able to resolve all the subunits of the minimal COMPASS complex that is sensitive to ubiquitin. And we were also able to determine where ubiquitin sits on this complex. And importantly, many parts of the COMPASS core complex were actually at quite high resolution, and the only parts that were at very low resolution were these very peripheral subunits here. And COMPASS is a very large protein, and when it binds to the nucleosome, it binds to one side of the nucleosome and actually spans the entire diameter of the nucleosome and takes up most of the surface here. So you can see that from the top, where this is the nucleosome here, and then COMPASS is essentially taking up the entire surface of the nucleosome. So if we zoom around the COMPASS complex, we can see that in the Set1 active site, the Set1 active site contains both the SAM cofactor and also an H3 peptide that surrounds H3K4. Now this H3 peptide is connected to a histone H3 molecule in the nucleosome. But when we saw this, we immediately noticed that this H3 interaction is completely different from that of Dot1L. So in the case of Dot1L, Dot1L recognizes ubiquitin on one side of the nucleosome and then directly methylates H3K79 on the same side of the nucleosome. However, for COMPASS, we can't see any density for the residues between the H3 residues in the Set1 active site and the H3 in the nucleosome. But when we compare the distance between the H3 residues in the Dot1L active site with the copies of H3 in the nucleosome, you can see that the H3 in the Dot1L nucleosome, in the Set1 active site is much closer to the histone H3 copy that is on the opposite side of the nucleosome. And so what we think is going on is that COMPASS actually recognizes ubiquitin on one side of the nucleosome, but ends up methylating the histone H3 copy on the opposite side. And this really highlights that the same mark, the histone H2B mark, can template histone methylation either in a cis fashion with Dot1L, where it's methylating the same, the H3 copy on the same side of the nucleosome, or in a trans fashion, just depending on which mark is being deposited. And this also highlights that even though nucleosomes are highly symmetric complexes, there can actually be a very high degree of asymmetry in regard to the modification landscape of these histones and how these histones are read out and decoded by the cell. So in addition to showing how COMPASS interacts with Histone H3, the structure of the nucleosome bound COMPASS showed that COMPASS actually changes conformations upon binding to the nucleosome. And shown here is just the published structure of COMPASS in isolation here by the Skiniotis lab. And then if you just pop in a H2B ubiquitinated nucleosome here, and this is the H2B linked ubiquitin here, you can see that when COMPASS binds this nucleosome, it actually flexes around its midpoint and brings this part of the complex down to contact the nucleosome. And the result of this conformational change is that COMPASS directly interacts with nucleosomal DNA on both sides of the nucleosome. On one side, this DNA is docked into a positively charged channel between this BRE2 subunit and Set1 catalytic domain. And on the other side of the nucleosome, a positively charged but unstructured loop in this SPP1 subunit directly contacts the DNA. And in addition to the contacts with DNA, the conformational change brings this SWD1 subunit directly in contact with the histone core. And finally, a long arginine-rich sequence of Set1 that is disordered in the isolated COMPASS forms a long helix that directly interacts with the histone core. And this helix was very exciting because prior literature from the Rader lab in 2013 had shown that this arginine-rich sequence of Set1 is critical both for the activity of Dot1L on nucleosomes and also for its ubiquitin-dependent stimulation. So just as I mentioned, this RxR helix is disordered when COMPASS is not bound to the nucleosome, and then becomes structured only when COMPASS binds the nucleosome. And when COMPASS binds the nucleosome, this RxR helix actually passes underneath the entire complex and snakes up back here to the top of the complex. And this position of the RxR helix allows this part of Set1 to interact both with the H2B linked ubiquitin and with the H2A H2B acidic patch. So you can see that here, if we look at one side of this RxR helix, you can see that there are multiple conserved arginines that make direct electrostatic interactions with all of these acidic residues in histone H2A. And importantly, mutating any one of these arginine residues has very dramatic effects on the ability of COMPASS to methylate histone H4, histone H4. And on the other side of this RxR helix, a pair of hydrophobic residues at the tip of this helix actually directly interact with the H2B linked ubiquitin within this hydrophobic pocket that is formed by the same residues that are used by Dot1L. But in this case, these residues are in Set1 and just a single, or these two residues come in here and just bind into this hydrophobic patch on ubiquitin. And this then actually explains why this RxR helix was critical for the ubiquitin-dependent stimulation of Set1. So in addition to the RxR helix, which only forms upon nucleosome binding, another part of Set1 actually changes conformation to enable the interaction with the histone core and with the ubiquitin. And so what we have here is this helix of Set1 in the isolated COMPASS structure. And if you just dock the isolated COMPASS structure into the EM density of the nucleosome-bound COMPASS structure, you'll see that there is this clash that occurs between this helix in Set1 and the histone core. However, upon nucleosome binding, this whole helix unravels in Set1, and new interactions are made between this part of Set1 and the histone core and with ubiquitin. In this case, a electrostatic interaction is produced between this R936 residue and E72 of H2A, and this L928 residue actually sticks up into this same patch of ubiquitin that I just showed on the last slide. And importantly, if you mutate this arginine 936, either to alanine or in a charge reversal to glutamate, in yeast, you completely abolish the ability of native COMPASS to methylate histone H4, either di or trimethylation. And finally, in addition to the contact between the RxR helix and ubiquitin, there's also a contact between ubiquitin and this other COMPASS subunit called SWD1. However, if you look at the isolated COMPASS structure where COMPASS is not bound to the nucleosome, the entire binding site for ubiquitin is malformed. There's no indication here that ubiquitin could bind here. However, when COMPASS binds to the nucleosome, this part of SWD1 completely rearranges. And the result of that rearrangement is that SWD1 presents a single hydrophobic surface that is then used by ubiquitin to dock onto this surface. And in particular, this cluster of hydrophobic residues in SWD1, this L12, V401, and F9, bind to a hydrophobic surface on ubiquitin consisting of isoleucine 44, valine 80, and leucine 8. And if you mutate just the center hydrophobic residue in SWD1, this leucine 12 to alanine, that mutation completely abolishes the ability of COMPASS in yeast. So native COMPASS, mutating this residue in native COMPASS completely abolishes its activity in the cell. So now that we have this nice structure of COMPASS bound to a ubiquitinated nucleosome, we can begin to understand, or try to understand, how this ubiquitin is actually able to activate this complex. And in this case, there are a few structural things that help us understand this. Importantly, ubiquitin helps to order this RxR helix, which is critical for Set1 activity. And this RxR helix is also important for ubiquitin binding. So that's one possible explanation for how ubiquitin is able to stimulate this complex. However, there was another structure of a different yeast COMPASS complex that was published by the Zheng lab that also included a structure of COMPASS bound to a nucleosome that did not contain ubiquitin. And the position of this RxR helix was more or less the same, and ubiquitin binding was not impaired. So I guess the jury is still out on exactly how ubiquitin is able to stimulate this enzyme. And finally, how can we compare these two different structures in terms of how they are stimulated by ubiquitin? So in the case of Dot1L, ubiquitin binding, yeah, sorry, I'm getting, sorry. Ubiquitin binding directly probably restricts the conformational search of the enzyme for its substrate. And then the distortion of the substrate is stabilized by all these other stabilizing interactions between histone H4 and also binding to ubiquitin in general. In the context of COMPASS, ubiquitin seems to favor conformational changes that stabilize this RxR helix onto the nucleosome. But that's with the caveat that even nucleosomes that do not contain ubiquitin also have this RxR helix binding. But also ubiquitin binding stabilizes COMPASS conformation for different conformational changes as well. So there's conformational changes that occur within Set1 that aren't the RxR helix that are also important for interacting with ubiquitin. Okay, so with that, I'll go ahead and end my presentation and thank people in my lab, especially Cynthia, for giving me great support in this postdoc, and also especially Nick and Chad and Changbin, who all helped me a great deal in my quest to determine all these structures. And with that, I'll take any questions.
Great. Any questions for Evan? You can raise your hand, and if you go to the let's see, is there a raise hand here? You can send me a chat. You can send me the question directly. We can unmute you and ask your question. I see we have a couple here. I can kick it off with just a sort of general question. There's a lot of conformational changes going on here. It's a fascinating molecular machine. I know there's also a lot of pieces sort of coming and going of like probably not particularly high affinity. In your 2D classes for your particles, is there any hope of like low populations of conformational states that might give you insight into like what these conformation, like how, you know, or is it just there's a lot going on, right? So
yeah, so I did not do an exhaustive search for different conformations with particle classification. It does seem that the huge majority of particles for both structures, both of Dot1L and of COMPASS, were primarily in one orientation. And that might be due in part due to the norleucine mutation that I prepared to make those complexes, especially in the case of COMPASS. I didn't show the data, but the norleucine mutant does actually increase the affinity of COMPASS for the nucleosome by a very large margin, like 10 to 50-fold higher affinity. And so I didn't do an exhaustive search, but it did seem like most of the particles were all in one state.
okay. All right. We had one from Pete Meyer in the group here, if I didn't take his question. Pete, did I take your question?
That was me clapping.
Oh, okay. Oh, it was clapping. Okay. Any other questions?
Yeah, I have a question.
Yep. And we had one from Jared Bard here. So Jared, we'll do Wolfram and then we'll come back for Jared.
Okay, so I think it's quite intuitive to assert that the deprotonation might be promoted by the hydrophobic environment in general, but this was a aromatic environment in particular with this tryptophan nearby. And that's kind of counterintuitive because I would have thought that the positive charge would be stabilized by that through a cation-pi interaction.
Okay, so this is getting a little bit far afield from my expertise. I will say that there are other hydrophobic residues other than the tryptophan in that pocket. So it's not simply a aromatic environment. There are a whole other set of hydrophobic interactions, just aliphatic side chains in that same pocket. Also, I don't have any information on what a lysine would actually look like in that tunnel because we only have the norleucine mutant. So it's very possible a lysine could make slightly different interactions, but we don't have the data for that. And yeah, we did consult with a physical chemist to look at, not look at the structure, but to ask questions about how this pKa might shift when you force this lysine into a hydrophobic environment, but we did not actually consider any kind of stabilizing cation-pi interactions.
Thank you.
Go ahead.
I had two questions about the Dot1L ubiquitin interface. The first is, it seems like the interface is not particularly large. Do you think that there are other interactions in cells that are required to recruit Dot1L to the ubiquitinated histones? And then the second would be, now that you've seen this novel ubiquitin interface, have you been able to find other examples of it in other proteins?
Right, so to answer your first question, so yeah, Dot1L is recruited to genomic loci using different partner proteins. So we did not test, for instance, what effects these other proteins would have on, for instance, the affinity of Dot1L for ubiquitinated nucleosomes. But I will say that most of the affinity of Dot1L for nucleosomes is not imparted by the ubiquitin interaction. It actually comes from this DNA binding sequence. And that interaction with the nucleosome is very tight, on the order of 50 to 70 nanomolar. So it appears that, yeah, the ubiquitin interaction is very weak, and it's probably important for Dot1L to be recruited to certain loci with these partner proteins. But once Dot1L is there, this ubiquitin is not really increasing affinity, and maybe it's just doing some of these other catalytic effects. And in terms of your second question, we actually did not look for structurally similar proteins in RCSB to see if there was some sort of new ubiquitin binding motif that could match this Dot1L structure. That's a great idea. I may actually do that, but I don't have any information on that.
Thank you.
All right, great. Well, with that, I think that was the last question. Evan, thank you very much for a fascinating talk, great presentation, fantastic work. Thank you very much. And thank you for gracefully exiting the building, and the first time you'll give a talk during a fire alarm. So
anything can happen. All right, thank you so much.
These are strange times. Great, and thank you everyone for joining, and we'll see you next week.
Thank you for hosting.